Hello everyone, this is Hasiba Asma. I'm a PhD student at Technology Health and Lab in State University of New York at Buffalo. Welcome to my poster that's about improved enhancer discovery in Drosophila and other insects. So enhancers or cis-regulatory modules control the spatiotemporal pattern expression of nearby genes, thus their identification is very important to understand gene regulatory network. We have previously developed an effective method for genome-wide computational enhancer discovery that's known as scrimshaw. As you can see this figure at the bottom of first column, that's the main framework of scrimshaw. It's a machine learning algorithm and it uses training sets composed of known enhancers. We construct these training sets using our RegSys database that has approximately 23,000 experimentally validated enhancers in it. So we train our scrimshaw using Drosophila enhancers, and trained algorithm then gives us prediction not just in Drosophila but also in other insects like holometabolists such as mosquitoes, beetles, and bees. Now we are working on a number of things to improve the ways to use scrimshaw. Here I discuss 3 different approaches for increasing scrimshaw's effectiveness that I currently am working on. So first of all, we developed this pipeline, PCR-eval, that you can see in the second column, for in silico evaluation of scrimshaw's prediction. It computes 3 measures of assessment. Basically, it compares the result of prediction with experimentally validated enhancers in red flag. And this feature is called red flag recovery. It also gives us the estimate and precision and sensitivity of the given method. Besides that, it can also evaluate sensitivity and specificity of specific training set that we use to train it. For instance, in column 3, the graph at the top shows aggregate and individual performance of different training sets that we used for our work. Their comparison with the random expectation has shown that most of our compiled training set performed better than random training set, and those random training sets basically are just chosen from non-coding sequences. In those few of the training sets that perform only equivalent or worse than random training sets are in need of reconstitution. And then our second approach is to use the comparative genomics in order to give individual scrimshaw predictions some sort of confidence score. For this approach, our assumption is that if our scrimshaw prediction is a true CRN, we should be able to predict it in multiple related species in orthologous scopus, as you can see in the figure at the bottom of column number 3. So incorporating such confidence measure in assessing scrimshaw output will be very powerful improvement to distinguish true positive results from false positive. And finally, our final approach is iterative searching. So evaluation has indicated some of our weak training sets. We took one of those weak training sets that has 7 enhancers in it and ran scrimshaw and got the predictions. Once we had the predictions, we validated 18 of those predictions. It had both true positive and false positive results. More precisely, 10 of them were true positive and 8 of them were false positive. For instance, in this figure in column 4, the middle one, KRH1, was a true positive and PDP1 acted as false positive. Then we reconstituted our original training set by adding only those 10 new enhancers in it, and reran scrimshaw on it. When we analyzed the result from this second round of scrimshaw run using updated training set, we found that it still had those 10 true positive that were previously validated as true enhancers in its top prediction, but now with this updated training set, it no longer had predicted those false positive as its top prediction. As you can see, this table summarizes this results. So these results suggested that using our updated training set, we have increased our true positive to false positive ratio, and thus we have markedly improved our precision specificity. So again, using these approaches, we are continuously working to find more ways to use scrimshaw effectively. Thank you so much for listening.